trees, which allows for a larger amount of hammocks because there are more places that you can attach the strings of the hammocks onto different types of trees. And on the bottom, there's only 2 trees, whereas you can only fit 1 hammock. This relates to our study because structure is very important to spiders. Spiders, like hammocks, have to be able to connect their webs to many different places to allow for the complexity necessary to build a web to catch the predator.
And with this physical structure of plants, it allows this mini attachment for web structures, which makes it secure. And these spiders in which we were studying can use this like stability in their web to help catch prey by sensing it through their vibration. And kind of like what the last group said, invasive species, they outcompete the native species, and especially with our study, honeysuckle. And it outcompetes them for sunlight resources and water and just many other aspects.
Previous studies similar to ours have been conducted. Loomis et al. In 2014 conducted a study on the effects of honeysuckle where it's present and absent. And when they conducted their study, they found that where honeysuckle is present, there is a much higher population of spiders as well as other insect species. So due to Loomis's research and other research, we've often been faced with deciding whether native plants or native animal populations are more important because invasive species such as honeysuckle will overrun a native plant population, whereas it is very useful for the spider populations as spiders need the structure to build their webs. So we are hoping that our research can provide more information as to whether or not you have to choose between the 2 species.
So here's just a picture of a live honeysuckle, and a honeysuckle has been foliar killed, and you can see there's quite a big drastic difference between. So our question, our experiment, is there a difference in species abundance, richness, and diversity of spider populations in the area where honeysuckle is living versus where it has been foliar killed?
And we hypothesize that in areas where honeysuckle is alive, there will be a higher spiders population, species diversity, and species richness versus in areas where it has been foliar killed and left standing. This is a picture of Lipton Conservation Area in Independence, Missouri. This is where we conducted our research. If you look on the left side of the path, that's where we conducted our research from the standing dead honeysuckle treatments, where the honeysuckle has been foliar killed, and to the right is where the honeysuckle has been left living.
So here's a picture actually at Lipton Conservation Area of the standing dead honeysuckle. Here's a picture of the standing dead honeysuckle, and behind it you can see that there's a living honeysuckle. And, okay, so for our active sampling of spiders, we went to certain sites like the standing dead site, and we would take this beat sheet and we would put it underneath the honeysuckle bush and this beat stick, and you would literally beat the bush until leaves and other leaves, spiders, sticks, whatever fell on to the beat sheet, and we would use this thing called a pooter, and you would literally suck up the spiders into a vial, which was kind of scary at times, not close to the spiders, and then after doing that, we would go from tree or bush to bush and beat it, and then in the vial that the spiders were collected in, we had to transfer it to another vial of ethanol quite frequently due to spiders making web attachments into the pooter and also starting to kill each other off. So we had to make that switch very quickly. And in our sampling, we had 6 samples at each site. So 6 for the standing dead treatment and 6 for the live treatment. And at each site, we spent an hour time of sampling.
Once we had collected those specimens, we took them back to the lab here and begun species identification. So in order to identify our spiders by species, we put them in a petri dish, and we filled the petri dish with ethanol, and then we put them under a microscope. And then, thank you. Dr. Kowinski helped us by mapping out common spiders that are here in Missouri. He gave us a list of common features and shapes and sizes of the spiders, and we also used the spiders of North America book to help us classify them. Once we had identified our spider by family and species, we put them in a smaller container also filled with ethanol, which we labeled with the time and date they were collected, who collected them, what treatment they belonged to, whether that was living or dead, who identified them, and what family they belonged to, and what species we labeled them as within the family.
For our data analysis, we first ran a Shapiro-Wilks test to test for normality in our treatments, and in the live sample, species abundance, diversity, and richness were all normally distributed, but in the dead sample, abundance and diversity were actually not distributed normally. And to test the difference between spiders, abundance, richness, and diversity, we ran a one-way ANOVA using R.
Just to recap, before we move into our results, we hypothesized that there would be a greater spider species abundance, richness, and diversity in areas where honeysuckle is living and dominating versus where foliar filled is left.
And here's our mean abundance for spiders, the x-axis being the types of honeysuckle that are standing dead or alive, and the y-axis being the number of spiders per hour sampling, and we have error bars using 95% confidence intervals, and from our one-way ANOVA we got a p-value of 0.09, which means that there is no significant difference between the two treatments, the dead and live.
Similarly to abundance, on our 95% mean confidence interval for species richness, we found no significant difference between the two treatments, and our p-value of 0.14 supports this.
And similarly again with our species diversity, we also, which was a p-value of 0.83, which results in no significant difference between the dead and the alive treatments. So, kind of going back to our hypothesis, our results were completely opposite of what we thought. We actually saw more trend towards species abundance, diversity, and richness in the staining dead honeysuckle compared to the live honeysuckle.
There are several biological reasons for why our results were so significantly different from our hypothesis. One of the problems could have been with where we conducted our research, because the living honeysuckle and the standing dead honeysuckle were in very close proximity, so this could have provided very similar food sources between the two areas. In areas where the honeysuckle is dead, it has allowed for native species to regrow, which will cause more ground litter, like leaf fall and dead grasses, which provides a good home for insects. And taking it back to the analogy, in areas where honeysuckle is living, there's a lot more extra branches and extra leaves, and if you've ever hammocked in a tree that is full of leaves and branches, it might be very hard to connect your hammock in areas where there's a lot of extra things, whereas for spiders, that may be the case to limiting the abundance of them in that area.
So for future studies, we would like to go to a site where the live and dead samples of honeysuckle are significantly separated from one another. So different different insects couldn't be in the same population of honeysuckle, because we thought there might be live honeysuckle insects getting onto our dead honeysuckle treatments and stuff, which could affect our results. And we would also like to look for trends in different morpho species where they are specifically more predominant in live honeysuckle compared to dead honeysuckle. And then taking it back to Lewis, Lewis wanted their management of honeysuckle was to keep it alive to give spiders population. But based on our results, we would like to start killing many more honeysuckle and leave it standing. And this will still give the population or the spiders a habitat to live in, and it would also give the understory a chance to regrow, since there will be more sunlight and more resources to get to the understory. And we would like to thank Professor Nickel with help throughout this whole project and data analysis, and Dr. Paul Kowinski with the help of identifying spiders, and the William Jewell College Biology Department with giving us the space and resources needed to fulfill this
Seth, to follow up on that, so in your study, so yes, there's this importance to structure, right? Having these sticks and branches and things like that for all these webs to build on. But the setup of your project in the both live and standing dead, that's the same. There's the same amount relatively. I mean, yeah, there's leaves and stuff maybe getting in the way, but there's relatively the same amount of structural support for webs. So why did you hypothesize that there would be more spiders in the living than the dead if there's the same amount of habitat structure available?
I would say food availability was probably one of the biggest factors, because if the plant's alive, it can support more insects, and I don't think considered the proximity of other living plants as well as the living honeysuckle part of the treatment.
If the spiders were all eventually euthanized, why were only some of them selectively euthanized in the collection?
They were all, as we collected them, we dumped them into the ethanol because they would kill each other and build webs if we left them in the
Okay.
You talked a little bit about doing about foliar killing honeysuckle. Could you? I know you talked a little bit about kind of what the purpose of it is, but could you go more into depth about like what it is?
It's kind of just like a herbicide in the killing species. So it kind of kills off the plants and the leaves like you should like. So here's a picture of standing there. There's a lot less leaves on it and stuff like that. So pretty much the foliar killing is just killing off the.
It's an herbicide
Yeah,
sprayed directly
herbicide.
onto the leaves, so
Yeah.
it kills it through the leaf system.
How does that compare to other methods of honeysuckle removal? Or a honeysuckle, sorry. When let's say you have a piece of land like Lipton Conservation Area or Martha Lafitte Thompson Nature Sanctuary, right? And you have a honeysuckle invasion, right? The idea generally is to try to remove the invader. How is that generally done, and how is it different at Lipton?
I think generally they completely remove the species, like there's no structure left there. They like completely uproot it.
Completely wipe out the part which takes a lot of hours and manpower to try to get them out. So they're trying to foliar kill it, which spray with chemicals, which takes a lot less time and stuff, and maybe the same results would produce in like understory growth and stuff, since there would be more resources available for the understory.
I had a question about your sampling. So, you said you sampled in an hour per site, and I guess you said you beat bushes, and then you said you sampled for an hour. So, like one sample, is that like multiple bushes and just determined by amount of time?
Yeah.
Okay.
Just amount of time sampling.
Okay.
So,
So does that mean you like sampled a certain number of bushes per hour, or it was just like how fast you're moving, how much structure there was there?
Yeah.
Okay. Which I know like time thing is common. I was just curious about what that actually meant. Can you go to your first graph, please? Okay, so number size per hour. What was your next graph? Okay. So you identified two morpho species. What does that mean?
We actually identified, it was data not shown, but we actually identified 13 families of spiders that we overall collected, and with each family we broke it down into their individual species that we saw them in. So I think the most was
About 4 different species. I think one family had about 4 different species, and that was the highest amount of species.
Okay, since
She's asking about what's the difference between a morpho species and a species.
so did you actually identify like the specific species for each thing?
we just gave the species a number.
We identified them by family, and then like we called them species 1, 2, 3, or 4 as we identified them. Like if they had similar characteristics to another
had similar morphologies? Oh, okay. Hint, hint.
I know what it is.
So you didn't actually identify species?
Specific
Okay,
type of
just
species.
based on like similar characteristics within a family.
Yeah.
What were some of the different families you guys found then? Like, what were the, so basically what I'm wondering is, what are some of the characteristics of spiders that you found? Like what families are they in, how big are they, what type of nest do they build, just general things about
Probably the
species.
most common family that I saw was Thomisidae. It's like, kind of like a crab-like spider.
Okay.
Usually spiders will walk, their legs go forward. Crab spiders are literally like a crab. They kind of walk out like this.
These are the kind of
Very,
little ones,
very,
right?
very, very small, and I'm surprised we found that many because they're. I'm sure we collected 290 total spiders, and I'm guessing over half of them was probably the species, the family Thomisidae.
And those crab spiders are known to make webs, like in sharp canopies. Are all the families you found known to be kind of web building canopy species?
That will have to be further research because I personally do not know if they all make spiders.
What did you look at with the families that you didn't present? There's some information.
All of the species, but 3 of them were found in both treatments. There are 2 species that were only found in the live treatments, and 1 species that was only found in the dead. So that's why in our further research we went to, if there's a greater separation between the treatments, see if certain species can only inhabit either the standing dead or the living, because we found such a similarity.
How far away from the boundary between the 2 habitat types did you sample?
Couple feet in, probably.
What?
There's like about a 10-foot like separation between them, and you probably would walk 3 or 4 feet in before sampling. So.
Yeah, so certainly like edge effects or something, right? So if you sampled like distance from edge of some boundary, that might be interesting to see if the similarity of species or morphospecies composition is changing kind of as you get further from the habitat.
So you guys can correct me if I'm wrong. My understanding is that the 2 areas where they sampled from were fairly distinct, but around the standing dead there were little islands of living. They didn't sample those islands, but those islands could still be affecting
Close enough,
the
yeah.
samples of the dead.
Yeah.
Does that make
Yeah,
sense?
yeah, definitely.
Was just one person beating them as a control, or were there multiple?
All of us.
Okay.
Oh,
How many people is that?
yeah, that's what I was just
We
gonna
had
ask.
3 people, and kind of like another future study. We did this throughout late October and early November, so maybe doing this in another season, I guess, will maybe produce more spiders.
So were 3 of you sampling every time you went out?
The first time it was me and Alexis, and the second time was all 3 of us, and then the last time was
And
me
so
Alexis.
both times you sampled for an hour?
We sampled an hour per treatment. So the very first day I spent an hour in the living honeysuckle, and he spent an hour in the dead. And then the second day, all 3 of us spent an hour in each treatment. And then on the 3rd day, we each spent an hour in each treatment. Just Colby and I.
So I guess what I'm getting at is, was your per person effort the same each day? Because if you sampled the same amount of time but with more people, that would equal greater sampling effort. So based on how you just described that, that's not what happened. Okay.
We don't know for sure if our effort was the same throughout. I mean, but that could be something in the future study, like one person specifically does the treating, collecting of the spiders and stuff, just to keep that variable constant.
Yeah, no. So I guess when we, in ecology, we went out and sampled for earthworms and the leaf litter. And so we were supposed to sample a total of like 60 minutes of searching. Not 60 minutes. I don't remember what it was. Maybe that's right. But basically, if we had more people searching, each person could search for less amount of time to get like the same number of person minutes of work. So I guess what I was getting at is, were your person minutes of sampling equal on each day? So like, did you? And maybe it depends. Did each of you do a sample individually, or did you work together to get a sample? Okay. Well, then you do okay. I guess I was imagining like 2 of you collected one sample one day, and 3 of you collected a sample the next day. And in that case, you would have 3 people beating bushes for an hour versus 2 people beating bushes for an hour. So in this case, your sampling effort would be greater than it was here. But that sounds like that's not what you did. But I was not understanding. Okay. Okay. How did you calculate diversity?
We use diversity.
What does that do? What information goes into this calculation?
Or maybe back up and tell me what's richness.
Sure, go through your graphs again. Go back to the first graph.
What is richness measure?
It's the amount of different species or families.
Not families. You were closer the first time.
What's it say in your graph? There you go. Okay, so richness is the number of morphospecies, and then you calculated diversity. What goes into that diversity calculation? Did you guys calculate this like in Excel or by hand, or what'd you do?
Professor Nicole.
Oh, okay. I guess I know what to ask about on the final exam. Because we did talk about diversity, and we did talk about some.
You don't have to know the formula, but
Yeah.
what's the difference between diversity and richness? Like, why are they given 2 different names? So if richness is the number of, we'll just call them species. If richness is the number of species, what is diversity a measure of? Variance? I don't know.
What's your first graph? Okay. So what does diversity take into account that richness does not?
The abundance of different species.
There you go, the relative abundance of the different species. Any other questions? Oh, okay.
I have a question. So you said that you think that the honeysuckle plants should be left after they're fully repealed. Do you think that having those in the habitat like dead is worth like having that space taken up so that native species can't colonize that area again?
I think in terms of spider population, it's more beneficial because when it's dead, it's not limiting the resources of the other plants. It is taking up some space, but it's not limiting the light and the water that it would be when it's alive. And it's like able to keep the spider populations intact.
So would you say that native plants should or should not be able to recolonize areas that have standing dead honeysuckle?
Say they should.
Yeah, we should.
Okay. And what would happen over time? Like, let's fast forward. So let's say native species are moving in, and think about what happens sort of naturally when you have, maybe you don't know about standing dead honeysuckle, but maybe you know something about standing dead trees, right? They're called snags, right? What happens over longer periods of time in terms of sort of restoration? What's going to happen? What's the habitat going to look like, let's say, 10 years from now?
The state that will be there.
Why not?
Because it's weaker. It'll just like get overrun by the other plants.
Okay. And so will it just be barren then? I mean, I know you're not saying that, but.
Will the spiders still have a place to make their webs?
Potentially not. I mean, I think they can still make them in native species, but if the structure isn't there for the dead honeysuckle, it might create a problem for them if they're dependent on it while it's there.
So you said that the standing dead would be replaced by native species. If that happens, would spiders have habitat to make webs?
Say yes. I mean, it might not be nearly as sufficient, but it's still a structure.
It's not gonna be identical, right? 'Cause it's
Yeah.
mixed species, but is that what you were getting
Yeah.
at?
Yeah. So as new stuff comes in, it's going to replace and provide new habitat structure. And then as the standing dead starts, decomposing, breaking off in storms, whatever, that's going to go away. But then you have all this other new structure that's there for spiders. So you don't have this big crash. It's kind of like this, it's a smoother transition in restoration.
One more question. So you said the foliar killers when they use chemicals to like kill plants. Do you think that had like I don't know how much you researched about that, but like do you think that using those chemicals would affect any of the surrounding species more rather than just uprooting the whole plant?
I personally do not know.
I don't know how
I'm
it would
just
affect
curious.
the native species.
Think about the big area, like throughout the semester, where like at the very beginning of the semester, you saw big stands of honeysuckle. What was under them? What?
Not much.
Not much. So if you're spraying, what are you killing? Right. Are you killing other stuff? No, because it's not there. Right? So there's probably not as many detrimental effects to other plants as long as it's not a really windy day, right? And it's spraying up into the canopy or anything like that.
I just used to be because it'd be spraying on you as the sprayer.
Yeah, yeah.
So,
So they have to think about when they do it.
Yeah.
But you're pretty much just killing honeysuckle because that's pretty much all that's there.
And then I don't know if we know, I'm sure someone knows, the effects on like insects and other things. So you could be impacting